Plants transport water and mineral ions from the roots to the leaves. Plants move mineral ions from an area of low concentration in the soil to a high concentration in the root cells. What process do plants use to move these mineral ions into root cells? Tick one box. Let's have a look at the processes. Active transport is a movement of substances from a low to high concentration and requires energy. Now this is what's happening here. Substances are moving up their concentration gradient. Therefore, active transport is our answer. Now let's check through the other possible answers to ensure that we're correct. Diffusion is the passive movement of substances down their concentration gradient, which does not occur here. Evaporation is the conversion of a substance from a liquid to a gas, which also is not happening here. And finally, osmosis is the passive movement of water from an area of high concentration of water to a low concentration of water. Since we're not discussing the movement of water, this is not the correct answer. Okay, now we're being asked to describe how water moves from roots to the leaves. Okay, this is a describe question, so we need to state what is happening rather than explain. Notice here that it's a 2-mark question, so we need to make 2 clear points. So let's think about how water moves up the plant from the roots to the leaves. Well, it moves by transpiration, which is the process where there is an active pull on the water upwards. So this will be our first point, that it is how. So by the transpiration stream. Now a second point is going to be about where this occurs. So if we think back to the vessels in a plant, we know that the xylem carries the water. So the second mark here is going to be for mentioning that it's in the xylem. Plants lose water through the stomata in their leaves. The epidermis can be peeled from a leaf. The stomata can be seen using a light microscope. Now, table 1 shows the data a student collected from 5 areas on 1 leaf. Let's have a look at the data. So we can see it has a number of stomata in areas of both upper and lower surface, as well as a mean. Right, so what does the question ask us? It asks us to describe how the student might have collected the data in table 1. So, as it said in the question, the epidermis can be removed from a plant. Therefore, to see the stomata, we want to mount this epidermis on a slide and then use the light microscope that it describes. And it clearly says here that the number of stomata are counted in 5 different areas. So we're going to first tell the examiner we're going to count this number of stomata in 1 area. And then after that, we're going to repeat in 4 more areas to get a total of 5. As we've noticed before, there's the mean column in the data. So mentioning that we can calculate mean is a key thing to say. Now let's have a reference back to the mark scheme, and we can see that if I mark on in purple here, the marks that you would have got, there are already 4 marks from these very simple sentences. And it's therefore clear that it's very good to be concise in biology questions. You could also have said that by repeating the method in another side of the leaf to get another mark. Right, the next part of the question asks us what is the median number of stomata on the upper surface of the leaf. So let's have a look back to the previous table. And we're looking for the upper surface of the leaf. So to find the median, we need to write out the data in numerical order from smallest to largest. So let's do that here. And then in order to find the median, we need to find the middle value. Now to do this, we use the formula n plus 1 over 2, where n is the number of data points, in this case 5. This is the median. So 5 plus 1 over 2 equals 3. So we're going to use the third value, and that will be the median. So in this case, that is 1. So now let's fill that in on our answer. Calculate the value of x in table 1. Give your answer to 2 significant figures. Okay, let's have a look back to the table. So x here, as you can see, is the mean value of the lower surface for the number of stomata. So to find the mean, we want to add up all the numbers of stomata at each place, first of all. which comes to a total of 206. And so this number represents the total number, and we want to divide by the number of each individual number of areas studied, which is 5, to find the value at each on average. So 206 divided by 5, the number of areas studied, is equals 41.2. Now that is our answer. But this is a 2-mark question, so we must include some working in our answer. We have to be careful with the way in which the question is worded, and it clearly states, as we can see here, for 2 significant figures. So looking at our answer, it is given to 3 significant figures. So we have to change it so that it is into 2. So to find the answer to 2 significant figures, we look at the second number, which in this case is 41, and it's followed by a 2. Now since 2 is less than 5, we round down to 41, and that will be the answer to 2 significant figures. The plant used in this investigation has very few stomata on the upper surface of the leaf. Explain why this is an advantage to the plant. Now since it says explain here, we have to give the reason behind something happening. So a reason why this an advantage is so. So when we just think about the stomata, their role is to control the flow of water through the plant. Now, a plant needs water for its biological processes, so it's in its best interest to conserve water. So by having fewer stomata, it means that less water will escape. So that's the advantage, less water lost. But now we need to explain the reason why this is beneficial to the plant. And this is because it stops the plant wilting, so it does not wilt. Wilting occurs when the plant has insufficient water, and it's what makes a plant go droopy and often die. Tobacco mosaic virus, TMV, is a disease affecting plants. Figure 1 shows a leaf infected with TMV. Okay, let's take a look at the figure. Clearly here it shows a leaf, and we can see some discoloration, and it says the yellow patches are where TMV has destroyed chloroplasts. Now we need to take this into account because it will definitely come in handy later. Let's read the first part of the question. All tools should be washed in disinfectant after using them on plants infected with TMV. Suggest why. Now this is a 1-mark question, so we need to have 1 point. Now let's think about this question. With plants infected with TMV, there is likely to be viral particles of TMV. Now, in order to protect other plants, we need to try and prevent the transmission of this virus to other plants. Therefore, if it was on this plant and it got contaminated upon your hand or any equipment and it was then used on a subsequent plant, the virus would spread. So to prevent this, we need to kill the virus, and that is what a disinfectant does. So that's our answer, to kill a virus. Now that would have got a mark. And another thing that would have got a mark would have been to prevent the spread of the virus, which is essentially what we're trying to do by killing this virus. Scientists produced a single plant that contained a TMV resistant gene. Suggest how scientists can use this plant to produce many plants with a TMV resistant gene. Now, to create a plant offspring which definitely has this gene, we want to create a clone. Now to do this, we want to use a cell from a plant which can differentiate into any other cell, such as a stem cell. Now stem cells in plants are found at the tips of the roots, and this is known as meristem tissue. So what we're going to do is we're going to take a sample from this meristem, and that will be our answer. Alternatively, instead of saying meristem, you can mention tissue culture, which would also have got the mark. Some plants produce fruits which contain glucose. Describe how you would test for the presence of glucose in a fruit. Now, this is a food test, but we can't just test, for example, an apple or any fruit as a whole thing. So what we're going to do is we're going to create a paste. To do this, we're going to take a little sample of it, like a slice. I'm going to grind this down in a pestle and mortar until it creates a liquid. Now, this is the first stage, so let's write this down. Following this, we're going to add what's known as Benedict's reagent. Now this is initially blue, and we're going to add it to the sample, and following this we're going to heat it in a water bath. Now, if glucose is present, then the solution will turn from blue to a purple, So, to structure our answer, we've already begun to describe the method, and will continue to do so, by saying add Benedict's reagent and heat in a water bath. And finally, we need to describe the appearance of the solution, by showing what a positive result is. That is, what happens if glucose is present. In this case, it will turn from blue to purple. TMV can cause plants to produce less chlorophyll. This causes leaf discoloration. Explain why plants with TMV have stunted growth. It's a 4-mark question, but this question is actually level marked. However, it's still good to aim for 4 clear, concise points. Well, let's have a little think about what chlorophyll does. Chlorophyll is a green pigment found in plants. This is why, when there's less of it, it causes leaf discoloration. Now, chlorophyll is essential in the process of photosynthesis. So if there's less chlorophyll, there must therefore be less photosynthesis occurring. That's our first point. Now, photosynthesis is a process which produces glucose. So, with less photosynthesis occurring, there is also less glucose being produced. Growth is a process that requires lots of energy. So energy is very important. Now, glucose is used in respiration, which is a process that produces energy. Therefore, less glucose means less energy will be released for growth. Now, for these last 2 points, there is an alternative way we could go about getting the marks. Proteins make up much of all living things, so they're of high importance for growth. Now, glucose is required to make proteins, because it's a very energy-intensive process. So we can mention this, and we could also say, since there is less glucose produced, there will be fewer proteins. Now, these are essential for growth, so it's vital to mention that. Microorganisms cause infections. The human body has many ways of defending itself against microorganisms. Describe 2 ways in which the body prevents the entry of microorganisms. So to prevent the entry, we're talking about primary barriers. So defense mechanisms the body has on the surface of the body. So, first of all, in the stomach, any food that's ingested experiences very acidic conditions due to hydrochloric acid in the stomach. So that's the first point. Acid in the stomach kills any pathogens from the food that one eats. Secondly, the largest organ in the body, the skin, protects your body by covering it and forming a barrier. So the skin forms a barrier which prevents the entry of microorganisms. And skin also has other aspects, and you could have also mentioned in the mark scheme that it secretes antimicrobial fluid. Now, there are other things we could have mentioned, which I have not mentioned here, and these include the fact that the trachea has mucus which traps pathogens. Alternatively, you could have said that bronchi have cilia which waft this mucus to the throat to be swallowed. And finally, as a separate point, you could have said that hairs in the nostrils trap pathogens. In 2014, the Ebola virus killed almost 8,000 people in Africa. Drug companies have developed a new drug to treat Ebola. Now, since we're dealing with a new drug, we have to assume that it could be dangerous and it may not work. Therefore, before it is implemented, we need to carry out several tests. Now, looking at this question, it's a 6-mark question, but in this case, it's level marked. So the examiner is looking for the quality of your scientific writing skills. But we still need to be sure to make clear, concise points. So the first stage in testing of a new drug needs to involve cells which are not part of a human body, since we don't know whether it's dangerous. So these are preclinical trials. And that's the first thing we'll do. We'll carry out preclinical trials to test for efficacy, dose, and toxicity. That is, whether it works, trying to find the dosage, how much would be ideal for a human, and toxicity, whether it's harmful to these cells. Once we know that it's safe on cells, we then need to test for its toxicity on humans. Something that's safe on cells may not necessarily be safe for humans. So we need to do clinical trials on healthy volunteers at low doses to monitor safety. And following this, knowing that it is safe, we're going to test for optimum dosage and efficacy. And this will be done in the form of a double-blind trial. Now, a double-blind trial is that where there are two groups, one given a placebo and one given the actual drug. Now, the use of a placebo accounts for the placebo effect, in which a participant given any pill or injection, regardless of it containing an active ingredient, will see a benefit due to the psychological effect of taking it. So, we need to describe a little bit about what this double-blind trial is so that we can get some more marks. So we're going to say that this uses a placebo that contains no active drug, and we're also going to talk a little bit about the scientific method. So there should be random allocation of participants to either group. Now we want to also mention the blind aspect of the trial. That is, that neither the patients nor the doctors should know who is in which. Now, once we've completed this trial, we need to check that the data is valid. So the data should be peer-reviewed, and this should help prevent false claims. So looking for our answer, let's highlight the key parts. We're talking initially about preclinical trials for efficacy, dose and toxicity. Then clinical trials on healthy volunteers at low doses for safety. Following this, we have to test for optimum dosage and efficacy. Do this in the form of a double-blind trial using a placebo that contains no active drug. There should be random allocation of patients to each group, and neither patients nor doctors should know who is in which. Once completed, the data should be peer-reviewed to help prevent false claims. So, the key things to include here are what we are testing for, how to carry out such tests, and a justification for why we do these in such a way. All living cells respire. Respiration transfers energy from glucose for muscle contraction. Describe how glucose from the small intestine is moved to a muscle cell. Now it's a 2-mark question, so we have to make 2 clear points. So glucose moves through the body, through the blood, dissolved in blood plasma. But the glucose here starts in the small intestine, so how does it get to the blood? Well, the first part of our first point is that glucose moves into the blood, but that's not sufficient detail. We need to mention the process by which this occurs, and this is diffusion. So glucose moves into the blood by diffusion. Now once in the blood, it needs to get to the muscles. Now we can mention that the blood transports the glucose, but once again we need more detail. So to include more detail, we're going to mention the blood vessel through which it moves. It's delivered to the muscle cells by capillaries, the smallest type of blood vessel. They are so small that substances dissolved within them can easily diffuse from them into the muscle cells that surround them. Figure 2 shows an experiment to investigate anaerobic respiration in yeast cells. Now, anaerobic means respiration without the presence of oxygen, and that's a key thing to note here, especially as it's in bold. So, let's have a look at the first part of the question. What is the purpose of the liquid paraffin in tube A? Tick 1 box. Okay, let's have a look back at the apparatus. Now, the liquid paraffin here lies atop the yeast cells in sugar solution. So, since we are trying to investigate anaerobic respiration, we're trying to prevent any oxygen entering the solution. Now, since oxygen is in air, the paraffin, therefore, is preventing the entry of air. So that's our answer. The indicator solution in tube B shows changes in the concentration of carbon dioxide. The indicator is blue when the concentration of CO2 is very low, green when CO2 is low, and yellow when the concentration of CO2 is high. What colour would you expect the indicator to be in tube B during the maximum rate of anaerobic respiration? Well, let's have a look at the apparatus again. Any gas that's produced by the yeast will move up this tube and enter the indicator. So, what is produced during the anaerobic respiration that's taking place in the yeast? Well, the equation is glucose converted by enzymes into CO2, carbon dioxide, and ethanol. So this would result in an increase in the concentration of CO2. And as we can see from the descriptions above, when the CO2 concentration is high, it will turn yellow. Therefore, our answer is yellow. So just how the experiment could be changed to give a reproducible way to measure the rate of the reaction? Include any apparatus you would use. Well, we know that rate is the same as the change in concentration or volume over time. Therefore, to work out rate, we need to be able to measure both the change in something and time. The question mentions reproducible, and this means any scientist could carry out the same investigation and get the same quantitative results. Well, we know here that from the previous part of the question that CO2 is produced, so we can measure this. The best way to measure this is to use a gas syringe, which shows the volume of gas being produced. Now, as we've previously mentioned, we also need to know the time. So we're going to use a stopwatch and measure the amount of volume in a given time for each experiment. Instead of using a gas syringe, we could have also used a measuring cylinder. To do this, we would invert the measuring cylinder in a basin of water. We would then connect a tube to the mouth of the measuring cylinder directly from the tube in which the yeast is respiring. This measuring cylinder would initially be full of water, so that any gas produced would displace it, thus measuring the quantity produced. Compare anaerobic respiration in a yeast cell with anaerobic respiration in a muscle cell. Well, first of all, let's begin with similarities. Both of these processes produce energy, but compared to aerobic respiration, they produce a small quantity of energy. Therefore, we can say they release small amounts of energy. Now let's move on to the differences between them. During this process in yeast, carbon dioxide is produced. This does not, however, occur in muscle cells. Another difference is the other products. In yeast cells, the anaerobic respiration process produces ethanol. This is often utilized in fermentation, such as in the production of beer and wine. However, in muscle cells, lactic acid is produced. This is the substance that causes muscle fatigue and makes your muscles hurt during exercise. This is our second difference. It's important to note here the comparative statements used. For each mark, you have to mention both yeast and muscle cells to get it. A student investigated the effect of different sugar solutions on potato tissue. This is the method used. Add 30 centimeters cubed of 0.8 mol per decimeter cubed sugar solution to a boiling tube. Repeat step 1 with equal volumes of 0.6, 0.4 and 0.2 mol per decimeter cubed sugar solutions. Use water to give a concentration of 0.0 mol per decimeter cubed. Cut 5 cylinders of potato of equal size using a cork borer. Weigh each potato cylinder and place one in each tube. Remove the potato cylinders from the solution after 24 hours. Dry each potato cylinder with a paper towel. Re-weigh the potato cylinders. Table 2 shows the results. Now, let's have a look at the first part of the question. Calculate the value of x in table 2. Well, as we can see here, x is the percentage change for the 0.2 concentration. Now, to calculate percentage change, we do the change, which is the same as final minus initial, which has already been calculated for us here, divided by the initial value, multiplied by 100. This is a 2-mark question, so it's vitally important that we show some working. So, using the formula here, we take our change in mass, 0.15, and we divide by the starting mass, 1.35. So we'll write this down, and then we'll multiply this whole answer by 100. This gives us a value of 11.1 recurring. Now, for this question, it's sufficient to give 3 significant figures, so our answer is 11.1. Why did the student calculate the percentage change in mass as well as the change in grams? Well, to answer this, let's have a look back at the table. Well, as you can see, the starting masses are all different. So, if we purely gave the change in mass, this would be unfair. As if 2 potato chips change by the same amount of mass, this may not be the same as a proportion of their initial mass. So by calculating the percentage change, this means there's no need to worry about the differences in the initial masses. It allows different potato chips to be easily compared without needing to worry about their initial masses. So to gain the marks in this question, we're going to say it's to allow the results to be compared. Or alternatively, you can say because the starting masses were different. Complete figure 3 using data from table 2. Choose a suitable scale and label for the x-axis. Plot the percentage change in mass and draw a line of best fit. So first of all, let's have a look at the x-axis. I have included the table here for ease. Generally, in science, the independent variable, that is what you change, goes on the x-axis. And the dependent variable, that is what is measured, goes on the y-axis. This can be seen here with percentage change on the y-axis. What we changed as an experimenter was the concentration of sugar solution. So that will be an independent variable and it will go on the x-axis. Well, our concentrations go from 0 to 0.8 mole per decimeter cubed. And as you can see on the bottom, there are 5 different squares. 5 does not easily go into 0.8, so instead our scale is going to go from 0 to 1, and each of the larger squares will be 0.02 mole per decimeter cubed. So we should then label it accordingly. And our label will be the same as the label in the table, the concentration of sugar solution in moles per decimeter cubed. Then, using the attached table, we need to plot the points. Now it's very important to be accurate whilst plotting these points, since there is a narrow range which the examiner will allow, normally plus or minus only 1 square. Now, once we've done this, as the question states, we need to draw a line of best fit. This should be as close to all the points as possible. Now this generally means there should be the same number of points lying on either side of the line. As before, it's very important to be accurate, since there is a narrow range accepted for the answer in the next part of the question, which requires the line of best fit. Use your graph in figure 3 to estimate the concentration of the solution inside the potato cells. Well, the reason for the change in mass is due to osmosis. Now this is when water moves from an area of a high concentration of water to a low concentration of water through a semi-permeable membrane. Now this is what results in the change in mass. So at the point in which there's no change in mass, the concentration of water inside the cells will be the same as that outside the cells. Since the concentration of water at this point is the same inside and outside the cells, so must be the concentration of solutes. Looking at the graph, no change in mass occurs when the line of best fit goes through the x-axis. Now from our graph, this is at 0.52 mole per decimeter cubed. But the mark scheme would accept anywhere from 0.45 to 0.55 mole per decimeter cubed. The results in table 2 show the percentage change in mass of the potato cylinders. Explain why the percentage change of results are positive and negative. Now it's a 3-mark question, and not only that, it's an explained question. So we need to both state what is happening and also the biological reasons behind it, so why it is happening. In this experiment, the change in mass, regardless of whether it is positive or negative, is due to the movement of water. Water will always try and find an equilibrium. That is, water will always move to an area of lower concentration. This process, by which water moves through a membrane from high to low concentration, is osmosis. Let's have a look back at the graph first of all. We can see that up to where the line crosses the x-axis, that is, between 0 and 0.52 mole per decimeter cubed sugar solution, there is a positive change in mass. Now this means that water has moved into the potatoes. Whereas after 0.52, there was a negative change in mass. Now this means the mass has decreased due to water moving out of the potato cells. So we can first state the trend. So between a concentration of 0.0 to 0.52 mole per decimeter cubed, water moves in. And after this, between 0.52 and 0.8 mole per decimeter cubed, water moves out of the potato cells into the surrounding solution. Now this will get us 2 marks, and to get the third mark, we need to describe the process by which this occurs. And as I've already said, this is osmosis. So our final point is that in both cases, water moves by osmosis. Suggest 2 possible sources of error in the method given on page 16. Error is when there is a difference between the measured value and the actual value of something. So let's have a look back at the method and see anywhere where the results that we obtain could be different to the true value. Well, looking at the first point in the highlights, we can see that the sugar solution is in a boiling tube. Now, since this is in an open top, it's prone to evaporation of water. Now, if water evaporates, the concentration of sugar in the solution would therefore change, since there's less volume. And this would therefore change our results and could cause error. Also in the first line, it states the concentration of solution. Now, unless it's completely accurately obtained, this solution could be slightly different concentration, and this would undoubtedly create an error. Looking further down the method, we can see that the potatoes are dried with a paper towel. Now, if they were insufficiently dried, then water or sugar solution would remain on the outside of the potatoes. This would increase the size of the mass that's measured, which is a source of error. Finally, there is a balance used to both weigh and reweigh the potato chips. This balance will have a certain accuracy, such as to 0.01 of a gram. Now this could create a source of error, since the actual answer may lie between 2 0.01s of a gram. Now any of the errors that we've mentioned would be valid and would gain a mark, but I'm going to choose 2, the concentration of solutions and the accuracy of the balance. Figure 4 shows the human digestive system. Let's have a look at figure 4. It clearly shows a human with a mouth leading to a stomach, small intestine, large intestine, rectum and anus. Alongside this we can also see the liver and the pancreas. Now the first part of the question asks us to label the stomach and pancreas on the diagram. Well, the stomach is the kidney bean shaped organ on the right hand side here, which churns the food. The pancreas is located just beneath or to the side of the stomach, and it often has this wrinkly pattern as shown here. Many people suffer from stomach ulcers caused by a species of bacteria called Helicobacter pylori. The stomach is lined with a protective lining of mucus. Helicobacter pylori are acid-tolerant bacteria which can damage this mucus lining. Suggest how an infection with Helicobacter pylori might result in a stomach ulcer developing. Well, an ulcer is a damage to the stomach lining, where the acid of the stomach damages the cells. Well, if normally in the stomach when pathogens enter, they are killed by acid. So these bacteria are not. And this is what allows them to be damaging. This is our first point. But since the question says suggest, we need to suggest a biological mechanism. So since the bacteria remain in the stomach, they can, as it states in the question, damage the lining. So this would mean that the tissue of the stomach, normally protected by this mucus, is damaged by the acid itself, thus causing an ulcer. Alternatively, for this second point, you can say that HCl, hydrochloric acid, the acid in the stomach, damages stomach tissue. Helicobacter pylori can also cause stomach cancer. Describe how a person infected with Helicobacter pylori could also develop liver cancer. So if a person has this bacteria, they may get stomach cancer. It does not say that that bacteria directly causes liver cancer, so the only way that we can assume that liver cancer is caused by it is by it spreading from this initial tumour in the stomach. Well, if it causes a cancer, we need to bear in mind there are 2 different types. The first one is a malignant cancer. Now these types of cancer grow much faster, and often cells of them can break away and do what is called metastasize. This means they form secondary tumours elsewhere. Alternatively, it may be benign. in which case it's enclosed in a membrane, and therefore the tumour grows, but it does not spread elsewhere in the body. So, the only way they could spread from the stomach to the liver is if the cancer was malignant. And this is our first marking point. If the cancer is malignant, it may spread between organs. And the way it does this is by cells breaking off and moving through the bloodstream to different organs. So the method by which this cancer spreads will be our second marking points, through the blood. Finally, we need to say what will happen once this cancer moves to the target organ, in this case the liver. And we'll say that it forms a secondary tumour, and that'll be enough for the third mark. Gluten is a form of protein found in some grains. Describe the test you'll use to find out if a protein is present in food. So we're trying to find a food test. And for proteins, this is the Biuret test. So first of all, we take our food sample, and if it is solid, we'll have to grind it down, for example, in a pestle and mortar to create a liquid. Or if it may be a liquid, we can use it as it is. And what we do is, we add Biuret solution. Now, Biuret solution is blue. And there's no need to heat it or anything. We simply add it, and then it should turn lilac or purple if protein is present. Now, to structure our answer, the first part will be to describe what occurs, how we do the test, and that is that we add Biuret to the sample. Secondly, we need to describe the positive result, that is what happens if there is protein present, and that would be a change in colour from blue to purple. Celiac disease is a disease of the digestive system. It damages the lining of the small intestine when foods that contain gluten are eaten. When people with celiac disease eat foods that contain gluten, their immune systems form antibodies to gluten. These antibodies attack the lining of the small intestine. This causes inflammation in the intestines and damages the villi. Symptoms of celiac disease include poor growth. Suggest why a person with celiac disease might have this symptom. Well, the key thing to note here is the mention of damaging to the villi. Now, villi are small extensions of the small intestines along the whole surface, which give a very high surface area, and this allows maximum absorption of food, especially glucose and amino acids. Glucose for the release of energy and amino acids to build proteins. Now, damage to the small intestine will reduce this surface area. So that's our first point. Damaged villi reduce the surface area for absorption. Now, if there's less surface area for absorption, less absorption will occur in the same time period. Therefore, there will be less glucose and amino acids absorbed. That's our second point. This is a suggest question, so we need to try and ascertain a biological explanation for the process that results in poor growth. So let's think about this. If there are both fewer amino acids and less glucose being absorbed, they will have two different effects. We'll take the molecules one by one. First of all, with less glucose, less energy is released, since glucose is involved in respiration, a process which releases energy. So therefore, the transfer of energy from food to the body is reduced. Secondly, with fewer amino acids, less amino acids are available to build proteins, since amino acids are the building blocks of proteins. A gardener is looking at the plants in his greenhouse. Some of the plants have a disease. Give 2 ways the gardener could identify the pathogen infecting the plants. Well, often pathogens in plants exhibit visible symptoms. For example, this could be discoloration of the leaves, such as yellow patches. It could be wilting of the leaves so they become droopy. Or it could be the change to the colour of the veins on the leaves. Now, to know which disease it is from these visible signs, we're going to need to compare it to some images from a database, from somewhere where somebody has already described the disease. So we can compare photos from a gardening website. and this will be our first point. Now, alternatively, instead of a website, you could have said a gardening book or magazine. Now, sometimes different pathogens, can create the same visible symptoms. Therefore, we can never be exactly sure which pathogen has infected the plant. Therefore, we could send it to a lab for analysis, where they can test for the presence of a specific pathogen. Now, this will be our second part of the answer. Plants can become unhealthy if they do not have essential mineral ions. Describe the appearance of plants with nitrate deficiency and magnesium deficiency. Let's have a think about nitrate first. Well, nitrates are made into amino acids, which then become proteins. Now, proteins are essential for growth, since they make up much of an organism's body. So with less nitrate, and therefore less protein produced, the rate of growth will be stunted. So that's the effect of nitrate deficiency, stunted growth. Now let's have a look at magnesium. Well, magnesium ions are a component of chlorophyll. Chlorophyll is the green pigment involved in photosynthesis, which allows the plant to create sugars. Now, without this magnesium, there will be less chlorophyll made, and therefore less green pigment in the leaves, so they will also appear yellow. Plants need other mineral ions. Potassium ions are needed for healthy root growth. Phosphate ions are needed for healthy flowers and fruit. The gardener makes his own garden compost. The percentage of minerals in his compost was compared with 2 fertilizers he could buy. The data are shown in Table 3. Let's have a look at Table 3. Well, it has the percentage mineral content. Now this includes nitrate ions, phosphate ions and potassium ions. Alongside this, it tells us the cost in pounds per kilogram. This data is shown for garden compost as well as fertilizers S and T. The gardener buys fertilizer S. Explain why he chose fertilizer S. Now, this is an explain question, so we need to give biological reasons behind choices. Now it's 4 marks, so we need to make 4 clear points here. So we're going to start by labelling each ion with what it does. Well, from the previous question, we know that nitrate ions are essential for good growth of the plant. Phosphate ions are used for fruit and flowering, as it mentions in the question stem. And potassium ions are essential for strong roots. Now let's first look at the nitrate ion column. We can clearly see here that fertilizer S, with a percentage of 5, has the most nitrate ions. Now with the most nitrate ions, this will allow plants to have the greatest rate of growth. So this will be the first of our 4 points. S has the highest concentration of nitrate ions, so will result in the greatest growth. Let's look next to another column in which we can see S is the highest, and that is potassium ions. So since it has the highest potassium ion concentration, that must mean it will have produced plants with the strongest roots. So that is our second point. That S also has the highest potassium ion content, so it will produce plants with the strongest roots. Now it's important to notice that with each of the points we're making here, we are quantifying them. We are explaining them. So we're not only saying that it has the highest nitrate ion or potassium ion concentration, we are explaining why this is beneficial. Now let's have a look back at the table for the next point. Well, if we look at the cost, we can see that compared to the other fertilizer T, that can be bought from the shops, it is far less expensive, at approximately half the cost of fertilizer T. So this will be our next point, that S is cheaper than T. Let's look back at the table and the final column that we have not mentioned. Now that is phosphate ions. Now, as you can see, in this case fertilizer S is not the highest, with a percentage mineral content of 1.3 compared to 12 in fertilizer T. Now this means in terms of phosphate ions, it is not the best fertilizer to use. So we should mention this. by stating that it does not have the highest phosphate ion concentration or content. Now, if we think back to what phosphate ions are essential for, we know that they are used for flowering and fruiting in plants. So, if a gardener has a preference for a fertilizer with less of these ions, we could assume that he's not interested in growing flowers or fruit. Thus, this will be our final point, that perhaps the gardener is not interested in growing flowers. And that will be enough to get the full 4 marks. Lungworm is an infection. Lungworm can kill dogs. It is caused by a small worm. Figure 5 shows the life cycle of the lungworm. Now let's have a look at Figure 5. Well, first of all, lungworm reproduces in the dog and infects the lungs. The larvae, the young stages of the worm, are coughed up from the lungs and swallowed. This means we're going to the digestive system. Let's have a look at question part 2. Worm larvae pass out in feces and are picked up by snails. And 3, dogs touch or eat snails. This will be a different dog, so it will get spread around, thus increasing the number of dogs that are infected. Which type of organism is represented by the snail in the life cycle of the lungworm? Take 1 box. Well, if we look back at the life cycle, we can see that the lungworm, that is the parasite, is within the snail during part of its life cycle. By this occurring, the snail then transfers the worm between different organisms. This means the snail acts as what is known as a vector, that is, a carrier of disease that transmits it between organisms. So the worms will be inside the snail during this part of the life cycle. So, therefore, it is a vector. Let's have a look at the other answers to rule them out. Well, a fungus is a microorganism, so it cannot be the snail. A parasite, in this case, would be the lungworm. That is an organism which leeches off another organism by living inside it. And a protist is also a microorganism, such as that causes malaria. So just how the spread of the lungworm disease can be prevented. Well, let's have a look back at the life cycle. Now, to prevent the disease, we need to disrupt one part of the life cycle. To survive, the lungworm must repeatedly go through all stages of the life cycle. And during this process, it infects both dogs and snails. Therefore, by stopping one part of the life cycle, we will essentially be killing the lungworm in the long term, and also preventing their transmission into the dogs and snails. So, for example, we could destroy the snails. This would prevent the lungworm having anywhere to live during this part of their life cycle. Thus, they would eventually die out. Alternatively, we could look at the dog stage, in which the lungworm is within the dog. Now, we could treat or isolate the infected dogs. Now, if we isolated the dogs, this would prevent them producing feces, which would then be consumed by snails, thus preventing the life cycle of the lungworm. Furthermore, treating them would also prevent the transmission of the lungworm to its feces. Finally, we can see that it spreads by feces, and we could therefore educate the dog owners that they should be picking up the dog feces. This will prevent it being consumed by snails, thus preventing the spread of the lungworm. So, it is a 3-mark question, so we need to choose 3 of these points for our answer. First of all, I'm going to say to destroy the snails. Then, to isolate the infected dogs. And finally, to educate dog owners about picking up dog feces. Malaria is a disease spread by mosquitoes. Describe 2 ways to control the spread of malaria. Well, since malaria is spread by mosquitoes, that means mosquitoes act as a vector of disease. Therefore, the life cycle of this disease is similar to that of the lungworm. That is, it resides within the mosquito until a mosquito bites a human, in which case the malaria is transferred to the human. When other mosquitoes come and bite this human, they are also infected with malaria, and therefore it's a cyclical process. So as before, to prevent the transmission, and therefore prevent people getting malaria, we have to disrupt this life cycle. So we can either prevent the mosquitoes biting humans, and we could do this using a mosquito net, or we could stop the mosquito breeding at all, therefore preventing it spreading the disease. So these are our two answers. Figure 6 shows photographs of some animal cells at different stages during the cell cycle. Let's have a look at figure 6. Well, in A, the black lines that we can see in the middle of the cell are the chromosomes, and they are also visible here in B. In C, however, no chromosomes are visible, but the large central organelle that appears darker here is the nucleus. Let's have a look at the first part of the question. It asks, which photograph in figure 6 shows a cell that is not going through mitosis? Now, mitosis is the process through which cells divide, and during this process, the chromosomes are continuously visible. Now, as we previously discussed, C is the only cell in which the chromosomes are not visible. Therefore, C is the cell not going through mitosis. Describe what is happening in photograph A. Now, it's a 2-mark describe question, so we have to give 2 points about what we can see is going on. If we look back at A, we can see that it seems like 2 cells are separating. Now, what is actually happening is that at the end of mitosis, during what is known as cytokinesis, and both the cytoplasm and the cell membrane are separating and dividing. And in this process, they form 2 genetically identical daughter cells, as is the process of mitosis. So these are our 2 points. First of all, that both the cell membrane and the cytoplasm are dividing. It's crucial that we include both. And instead of saying this, you could alternatively mention the process of cytokinesis. And for our second point, we have to say what this causes, which is that it is creating 2 genetically identical daughter cells. A student wanted to find out more about the cell cycle. The student made a slide of an onion root tip. She counted the number of cells in each stage of the cell cycle in one field of view. Table 4 shows the results. Each stage of the cell cycle takes a different amount of time. Which stage in Table 4 is the fastest in the cell cycle? Give a reason for your answer. Well, if we look at the data, we can see that the stage with the smallest number of cells in it will in fact be the one that takes the least time. Now, the reason behind this is that all cells will continuously be going through the cell cycle. A stage which takes longest will therefore have the most number of cells going through it. And on the other hand, a stage which takes the least amount of time would have the least number of cells going through it. So our answer for this will be 4. And the reason for this is because there's the fewest number of cells going through this stage, for the reasons that we've just discussed. The cell cycle in an onion root tip takes 16 hours. Calculate the length of time stage 2 lasts in a typical cell. Give your answer to 2 significant figures. Well, if we look at stage 2, we see that there are 4 cells there out of a total of 36. And the proportion of cells in stage 2 will be the same as the proportion of time it takes in stage 2 compared to the whole cycle. So the sum we're going to do is 4 over 36 times 16, that is the number of hours in the whole cell cycle, times 60. And that equals 106.6 recurring. Now, to justify why we're doing this, let's have a look at each part of the sum. 4 over 36 is the fraction of cells in stage 2. This is multiplied by 16, the total length of the cell cycle, to find the length of time that stage 2 takes. And then we must multiply it by 60, since we are given the value for the length of the whole cell cycle in hours, 16 hours, but the answer is required in minutes, as we can see below. So to convert between hours and minutes, we must multiply by 60, since there are 60 minutes in an hour. It's important to note that it asks us to give our answer to two significant figures. So to do that, we look at the second digit, that is after the 0, and we see there is a 6 afterwards. Now this is greater than 5, so we must round up to 110 minutes. Bacteria, such as Escherichia coli, undergo cell division similar to mitosis. Figure 7 shows a growth curve for E. coli grown in a nutrient broth. So if we have a look at the graph, we can see that the y-axis is the log 10 number of live cells, and the x-axis is the time in hours. Initially, there is a phase with little growth between 0 and 1 hours. Then there is a large increase in the number of live cells during point P, until at Q it levels off. The first part of the question asks us what type of cell division causes this change in number of E. coli cells at P. Now, as you already said, at P there is a large increase. Now, bacteria divide by a process called binary fission. It's similar in many ways to mitosis, but it's slightly simpler, since bacteria have circular rings of DNA rather than chromosomes. So this is our answer. Suggest why the number of cells levels out at Q. Well, if we look back at the graph, we can see, as the question suggests, that it flattens off at Q. Now, reading back to the question, we see that the bacteria are grown in a nutrient broth. Now, this is essential, since for cells to survive and hence grow, they need many nutrients for all sorts of biological processes and molecules, such as proteins or DNA. Now, there is a limited amount of nutrients in this nutrient broth. So, at point Q, we can assume that nutrients has run out. And this is our first point. So, if the nutrients has run out, this means cells can no longer divide and cannot go through the cell cycle. Therefore, the cells die. Now, this is one way of getting the second mark. Alternatively, we can say that because of the lack of nutrients, the rate of cell growth equals the rate of cell death, because this would also result in no change in the number of cells. Explain how the human circulatory system is adapted to supply oxygen to the tissues and to remove waste products from the tissues. Now, this is a 6-mark question, but it's level marked. Now, to get the highest level, level 3, we need to make a detailed and coherent answer. To do this, let's start thinking about the human circulatory system. Well, the human circulatory system is a double circulatory system. That is, that blood is pumped through the heart twice on every journey through the body. A simplified version of the circulatory system is shown here. Now, in this system, due to the delicacy of the lungs, it is pumped at low pressure through the lungs, whereas in the tissues, it's pumped at high pressure. Now, this is advantageous because if it's at higher pressure, more blood will flow through the tissues in a given time. This will be our first point. It is a double circulatory system which allows higher blood pressure and greater flow of blood and so oxygen to tissues. Continuing to look at our diagram, we can discuss some of the vessels which carry this blood around the body. So first of all, we'll talk about the pulmonary artery, which carries deoxygenated blood from the heart to the lungs, or the alveoli in the lungs, where it absorbs oxygen. Now, this links to the first part of the question about supplying oxygen to the tissues. Now, to continue this, we can talk about how this is carried back to the heart by a pulmonary vein. this allows it to be carried on its journey, which entails being pumped through the aorta to the tissues by the heart. Now, we've discussed the gross structure of the human circulatory system here, so now let's have a look in more detail at a part of it, which will be the capillaries, the smallest blood vessels which are found within tissues. Well, a diagram is seen here, and there are several advantages of this. Well, first of all, oxygen is carried by the red blood cells, which are present in capillaries and in fact in all blood vessels, since they are a component of blood, and they contain no nucleus. This means there's the maximum space for the molecule called hemoglobin, which is what carries oxygen in them. Now, there are several adaptations of these capillaries which allow for optimum transport of oxygen to the tissues and removal of waste products from them. Well, in these capillaries, they have thin walls which allows for optimum diffusion of oxygen out of the capillaries into the tissues that require it for respiration, and of the waste products from these cells surrounding the capillaries into them. Now, an answer like this would certainly gain 6 marks, but there are other points that you could make. This include the high surface area of capillaries for optimum diffusion, and also the way in which the blood returns to the heart in veins with valves that prevent backflow. In fact, you can mention any correct point in this sort of question and you'll gain the marks. It's all about making your answer clear, relevant and detailed. Monoclonal antibodies are used to measure the levels of hormones in the blood. Pregnant women produce the hormone HCG. HCG is excreted in urine. Figure 8 shows 4 pregnancy test strips. Let's have a look at Figure 8. Well, it tells us here that a positive test result is when a line appears in the control window and the result window. A negative test result is when a line appears only in the control window. And an invalid test result is when no line appears in the control window. Well, the first part of the question asks us which test strip shows a negative test result. Now, from the descriptions that we've just read, we know that a negative test result is when a line appears in the control window only. So looking across the control window, or the control window of all of them, we can see that only 2 have a line in them. And of these, only A has a line in the control window only. So this is our answer. Monoclonal antibodies are used for pregnancy testing. Give one other use of monoclonal antibodies. Well, one of the major uses of monoclonal antibodies is that of cancer drugs. Cancer cells are those which divide uncontrollably and rapidly in the body. Now, this is due to mutation, and as a result, their surfaces have different textures. That is, the antigens upon them, a protein or other molecule that sticks out of their cell membrane, are different to normal cells in the body. Therefore, monoclonal antibodies can be used to target these antigens, to either block receptors, which would prevent the cell from growing correctly, or to carry a drug to these cancer cells only. Now, alternatively, these antibodies can also be used to diagnose cancer in a similar way as the pregnancy test kit. They can be used to identify blood clots in the body, or they can be used to test for other hormones in a similar way to the pregnancy test. All of these would be acceptable answers. Figure 9 shows the parts of a pregnancy test strip. Let's have a look at Figure 9. Well, first of all, urine is applied here, at the base of the strip. 2, there's the reaction zone. There are mobile antibodies here specific to HCG. These antibodies can move and have blue dye attached to them. 3, the results window. Immobilized antibodies specific to HCG are found here. And finally, there's a control window, where immobilized antibodies specific to the mobile antibodies from the reaction zone are found. Let's have a look at the first part of the question. The pregnancy test strip will show a positive result when a woman is pregnant. Explain how the pregnancy test strip works to show a positive result. So it's an explain question. So not only must we describe what is happening, but why, giving biological processes behind it. Now, it's a 6-mark question, and in this case we have to get 6 distinct points. So let's have a think about what is happening in the process. So for there to be a positive result, the urine that is applied will contain HCG. In the reaction zone, specific mobile antibodies with a blue dye attached will bind to this HCG. Now, urine will continue to move up the strip to the results window. Here, the HCG will bind to immobile antibodies found here. Now, since these HCG molecules have antibodies with blue dye attached, a blue line will be formed from all the molecules of dye. There are more HCG antibodies than there are HCG molecules, so they will continue up the strip to the control window. Here, they bind to a line of immobile antibodies specific to the initial mobile antibodies. This, once again, creates a blue line. So, let's use this information to write our answer. Well, first of all, we need to describe what is happening with the urine. So, our first point will be that as urine passes through the reaction zone. Now, what does happen in this case is that mobile antibodies bind to the HCG. So we're going to write this. Our next point will be that urine continues to move up the strip, and HCG binds to the specific immobile antibodies in the results zone, creating a blue line. Now, as we've already discussed, there are more mobile antibodies than there are HCG molecules. So, any antibodies that have not bound to HCG move up to the control window and bind to the specific immobile antibodies here. In a similar fashion to before, a blue line is created, so we need to mention this, saying that thus a blue line is also seen in the control window. Now this is sufficient for the full 6 marks. And let's have a look through our answer to see where the 6 individual points are, as noted with red ticks.